Cool. Okay. Yeah, we can get started. Well, welcome everybody. It's very nice to see lots of returning people and a lot of new people. In today's eoPERCS seminar, we will be having Mónica Carvalho from the Smithsonian Tropical Research Institute giving us a talk on the end-Cretaceous extinction and the early evolution of Neotropical rainforests. Before we get started, there are a few housekeeping things that we just wanted to go over. A couple of announcements. You'll have the talk, a moderated question and answers after the talk. And if you guys would like to stick around for a tea time with the speaker, please feel free to do that. That's going to be around 30 minutes at the end of the talk and the question and answers. And as always, don't forget to send your questions via chat to Questions eoPERCS host, which is going to be Chrissy this time. And with that, a few housekeeping rules. eoPERCS values the participation of all folks interested in the paleosciences. Please remember to abide by our code of conduct during today's seminar. We do ask that you mute yourself for the duration of the talk. And please feel free to ask questions by chatting in the chat box of Zoom. And then if there are any technical questions or issues, they can go to the questions at eoPERCS host. We also now have closed captions built into Zoom itself. You can use the CC button to show or hide them. And if you know anyone who is an outstanding or an early career research scientist doing active research in the eoPERCS community, in the paleosciences community, please feel free to nominate them at the website that's given. Right there. And then we also have a weekly feedback form that will be put into the Zoom chat. It was just placed there. Please feel free to also fill that out. That gives us information in terms of who's viewing the talks, and it also helps us just look at the weekly demographic of the talks themselves. Okay. This week's speaker is Mónica Carvalho, who is calling in from Colombia. She did her bachelor's in biology from the Universidad de Antigua in Colombia. And then Mónica came over to the US and did a master's in science in geological sciences from Penn State University and completed her PhD in botany at Cornell University on the East Coast. And now is a postdoctoral fellow at the Smithsonian Tropical Research Institute at Panama. So has kind of moved all around following the plant ecology and how leaves and ecosystems respond to changing climate. Today, we will be listening to her give a talk on the neotropics. And with that, Mónica, I will let you take over.
Okay, thank you very much, Natasha, and thank you very much for having me here. It's a real honor to get to talk to all of you guys. So is everybody seeing the presentation correctly? Okay. So basically, in a nutshell, what I'll be talking to today, or what I'll be showing you, is how it's the extinction at the end Cretaceous triggered an anomalously long period of low plant diversity in the neotropics and actually enabled the evolutionary assembly of modern lowland neotropical rainforests. So when I say neotropics, I mean lowland neotropical rainforests. So neotropical meaning New World tropics. And these rainforests are super important, not just because they are centers of plant and animal biodiversity, but they're also fundamental for how Earth works today. They are fundamental for terrestrial carbon uptake. They have a major control over hydrological regimes regionally, as they control up to 50% of regional precipitation. In doing so, they control a big bunch or a big part of cloud cover, which in turn is albedo and reflects a lot of sun radiation back into space. And overall, these ecological services that neotropical rainforests provide are absolutely fundamental for human health and current climate dynamics. So what I work on, and as Natasha mentioned, is I study leaf fossils and plant fossils in general to understand the deep time evolution of these neotropical rainforests. And particularly with my team, we've been studying for the last 20 years or so, what was the effect of the end-Cretaceous mass extinction on these neotropical rainforests. And obviously, to this crowd, it's needless to say that this was one of the most important events in Earth's history. So, a big asteroid about 10 kilometers wide crashed against the Yucatan Peninsula, so shallow marine waters, caused this massive explosion, massive instant vaporization of rock that shooted out into the air, and all this vaporized rock once again condensed as it cooled down. All this debris fell back onto the earth, causing this massive, almost like rain of fire that was recorded globally and that we can see globally in the fossil evidence of, or the sedimentary evidence of these microtektites, which are these little glass spheres that formed when all this vaporized rock was falling back onto the earth. And in here we can see, like the distribution of a pretty old and classic paper, I must say, distribution of these microtektites that are associated to the iridium anomaly. So actually to the event, to the moment in which this asteroid hit the Earth. And, the widespread distribution really shows us the outstanding impact, the extent of the ecological effect that this event had all over the Earth. And we tend to associate the end-Cretaceous extinction. We think about, giant vertebrates, most dinosaurs, non-avian dinosaurs going extinct, huge marine reptiles, ammonites, and overall 75% of life on Earth becoming extinct. But we don't really know too much about what happened to plants, or we don't really think too much about what happened to those primary producers in terrestrial ecosystems. And there is some work that has been triggered or has studied what were the effects of this extinction on plant communities. And one of the big distinctions that there is in terms of this extinction when we think about animals and plants is that there is no equivalent to, say, dinosaurs in plants. So there's no big clade of plants that became extinct or almost extinct during this time period. Nonetheless, there is a lot of extinction, and there is extinction due to a massive ecological catastrophe. So for plants, for terrestrial ecosystems, this event was an ecological catastrophe that occurred globally and that we can see in the fossil record. So, most studies have been centered on high temperate areas, Williamston Basin, so the western interior of North America, down in New Zealand, in Patagonia, there's some in Japan. And what we can see overall is that there is quite a variation in extinction rates. And these are regional extinction rates that are estimated based on pollen. And they vary from up to 30% over in the Great Plains of North America to down as 10% extinction rate in Patagonia. And even though we don't really know what causes this variation, one hypothesis is that it could be related to just sheer distance to the impact site. And as you can see, we don't really know much about what happened in the tropics, and that's because we don't have a thoroughly sampled fossil record. There is one place, this is from Gorgonilla Island of Colombia, so it's an island off in the Pacific of Colombia, and it has this really, really extraordinary deposit of a KPG section, but this is a deep marine section in which the microtechnites are beautifully preserved. Associated to the KPG, there's a fern spike just above the KPG, which corresponds or is consistent with that initial recovery of plants. The problem is that we don't have any plant fossil record below this interval, so we don't know how the forests were, or we didn't know what forests were before the impact or how they were after the impact. And overall, one of the reasons that we don't know or we didn't really know, it's because, the tropics are absolutely wonderful for studying plants, but they're terrible for studying fossils. And that's because everything is just covered by this green, lush vegetation, because there's high precipitation, soils are very deep. Tackling this. And for the last 20 years, we've been collecting plant fossils. So we used both pollen and leaf fossils. And that's because when we think about plant fossils, because plant organs disperse in different ways when they fall to the ground and degrade and become fossilized, they're actually showing us very different things. So on one hand, pollen provides a very regional scope of what plant communities were in the past. And they're also very good because they provide a nearly continuous record in which we can track diversity through time. We can track community composition at a very regional level. So what we did, or what we've been doing over the years, we collected pollen from 39 different sites in order to build a composite record that would span between 72 and 56 million years ago. So the whole of the Maastrichtian and the whole of the Paleocene. And all these different sites are exposed, are either coarse or exposed sections, and they are along the eastern cordillera, found along the eastern cordillera of Colombia. And our second approach was to use leaf fossils. And leaf fossils are a very good complement to that regional scope that pollen provides. And that's because they reflect local vegetation. So most of these deposits are either channel or floodplain deposits. And we looked at specifically 3 different floras. So one pre-extinction or late Maastrichtian flora, which is Guaduas, dates from 70 to 66 million years ago, probably closer to 66. And 2 post-extinction floras from the Cerrejón, found in the northern part of Colombia, and the Bogota flora, found in the central part of Colombia. And overall, we looked at over 6,000 leaf fossils that are representative from these 3 sites. Now, 3 floras collected in various sites, so various localities in which we collected. And as I mentioned, one of the difficulties of finding these leaf fossils in tropical regions is that everything is covered by vegetation. So what we do is that we actually go to mines. So either open pit coal mines or underground coal mines or siltstone mines. And because these mining activities have already removed all the vegetation cover or all the topsoil, they actually give us a very good exposure of rocks in which we can look for these fossils. This site in particular, it has a beautiful outcrop that's actually found in the south side of Bogota. So this is actually almost embedded within Bogota, which is Colombia's capital. And in these sites, we use, pickaxes, hammers, and we've been able to gather, over 6,000 different leaf fossils from which we've been studying what happened before, what were forests like before and after the extinction, what happened during the extinction. So what we found is when we looked at this composite section in pollen, we see that, there's time, number of taxa, we see that there is a 45% extinction rate just at 66 million years ago, which is consistent with high extinction rates due to the ecological collapse associated to the asteroid hitting in Chicxulub. This drop in diversity is correlated with a peak in extinction and no change in origination rates, which is also consistent with a sheer effect of extinction, not necessarily change in origination. And what we see is that associated to this extinction, there is a very distinct clustering in plant communities before and after the event, which is mostly associated to a change in the proportion of flowering plants that we find in these pollen assemblages. So before the extinction, these pollen communities were about 50% flowering plants, and the rest is a mixture of ferns and conifers, mostly of the eoPERCS family, so monkey puzzle trees. And after the extinction, these forests, these plant communities actually became fully dominated by flowering plants. So that proportion changed from 50% to up to 90%. Showing us a very distinct change in the composition of this major plant group. With leaves, as I mentioned, we can infer we were able to infer a lot of very detailed ecological information. And that's because with leaves, we have abiotic and biotic conditions that are actually translated into things or traits that we can measure in leaves. So due to leaf size and leaf shape tells us about precipitation and temperature. Isotopic signatures of carbon in leaf cuticles can tell us about canopy structure. We can know about specific plant composition, so not just flowering plants, but what types of flowering plants were living there. And we have evidence of plant-insect interactions via herbivory in these very detailed feeding marks. So what we found overall is that all three floras, so all three forests before and after the extinction, are consistent with the physiognomy of tropical rainforests, meaning that they were all dominated by these large leaves, mostly entire margin, untoothed, a lot of drip tips. And these are traits that are specific to warm and ever wet environments, such as tropical rainforests today. And this is also consistent with, the long-term trends of, warm environments across the Maastrichtian and the Paleocene. We found, however, that there was a very, very distinct signal in canopy structure. So one way that we can look into canopy structure, into this ecological aspect of forests in the past, is looking at delta C13. And this is because closed canopies have a closed canopy effect. And what happens here is that whenever we have a forest that has a closed canopy, there is a restriction in atmospheric mixing, meaning that the air that's in the understory doesn't really mix with the air that is up in the upper canopy. Because of soil respiration and because of this restricted atmospheric mixing, there is a consistent depletion of CO2, of carbon-13 in relation to CO2 that is found in the air that's kind of trapped inside the forest. And because there is this depletion and leaves are uptaking this CO2 for photosynthesis, these ultimately translate into a very pronounced gradient in the isotopic signature of leaves from the upper canopy and from the understory. That varies about an 8 per mil, 9 per mil difference across these leaf strata. So one thing we can do with fossils is actually take a whole range of leaves that correspond to the same type, the same morphotypes or same species of plants, and see what is the variation in their isotopic signature. And what we found is that there is a very wide variation in the isotopic signature of the Cerrejón rainforest, which is a Paleocene rainforest, a floodplain deposit of a Paleocene rainforest, which is actually probably reflecting, the understory or the leaf litter that's constantly falling from the trees and was rapidly transported in a flooding event, something like that, rapidly buried, and it became fossilized. Whereas in the pre-extinction forest, we see that there is no such range of delta C13 in leaves, indicating that there is not a canopy effect. There is not an effect. We can't really see an effect of that closed canopy structure that would eventually lead to that isotopic range in leaf carbon isotopes. This other Cerrejón site is actually a channel deposit, and the leaves that we find there are mostly reflecting leaves that are outside of the forest. So it makes sense that this site doesn't reflect that closed canopy structure. But in general, we see that this is a very profound ecological difference in terms of the structure of the forest, in terms of how the forest worked before the extinction and how it was after. And for over 10 years, we've also been looking at the specific taxonomic affinities of a lot of these fossils in order to understand not just, what was the difference. We know that there was a difference in the proportion of flowering plants, but we don't know what flowering plants were there. So with leaves, we've been able to, with leaves and other types of fossils, such as seeds, flowers, fruits, we've been able to identify a lot of the families, of the plant families that lived in the past. So these are mainly Paleocene. Those are the ones that we've been able to identify kind of most straightforwardly. We find representatives of Malvaceae, which is the mallow family. We have melastomes. We have Menispermaceae, so the Moonseed family. This one is represented in a high diversity, both in terms of endocarps, which are these fossils over here, various types of leaves. We have members of Araceae, of the Araceae family, so a very, very distinctive tropical family today. We have multiple members of Ericaceae, so the palm family. The palms were very diverse back in the Paleocene. And in the Paleocene, there's this one striking feature, and it's that we find tons of legumes. And one of the reasons this is important is because legumes today have a very important ecological role in tropical rainforests. So overall, globally, these plants they have a symbiosis with nitrogen fixing bacteria, and this nitrogen fixing bacteria kind of speeds up the availability of nitrogen for plants. And in tropical rainforests, there is a very tight correlation between productivity and the amount of legumes, of legume trees that you have. And in the tropics, legumes are these large trees. They're canopy dominant. Some of them are even canopy emergent. So it's a very, very key family. However, when we look at the fossils of the pre-Cretaceous, pre-extinction, late Cretaceous forests of the Guaduas Formation, we don't see, we really haven't been able to identify a lot of these plant families. And that's because most of the fossils that we find have a very general angiosperm look, so flowering plant look, but there are no really specific traits that we can associate to specific clades within the flowering plant. We have been able to identify members of the Rhamnaceae, which is this family over here, and Piperaceae, which is the black pepper family. So when we put together, the one thing we can do with all these identifications is actually compare, the floristic composition of at the family level, at the plant family level, between modern living neotropical rainforests and our fossil forests. One of the reason we can do this is because taphonomical work, previous work, has shown that litter samples, litter that's collected from the forest ground, actually represents it reflects up to 90% of the standing biomass in a 12.5 meter radius. which means that, all the leaves that we collect here, which is an analogous sample to the fossil sites we find, they're actually they're a very good representation of the standing biomass. And these studies have been done in living rainforests, in which, we can control for all the plants that are living around the catchment site, or where the leaf litter was collected. So what we did was take this same principle, assuming that analogous leaf litter sample and its relationship with the standing forest, and look at the distribution of plants in two living sites, one in Barrio Colorado, Panama, and Parque Macajaco in Colombia. So this is an Amazonian plot. And these sites are very important ecologically because they're permanent plots, meaning that every single tree that has a radius over 1 centimeter has been tagged, has been identified down to species level, and is constantly monitored every few years to see how much these plants grew, which plants died. So it's a very powerful source of information for spatial distribution of trees. So what we did was kind of randomly select areas or small quadrants of both of these sites to create a distribution of how similar the plant family composition is within these forests, and we compared these forests with our fossil sites. So what we found is that, so here CDI is a measure of ecological difference, so difference in plant composition. 0 means it's a dissimilarity index, so 0 means high dissimilarity or low dissimilarity or high similarity, and 1 is very high dissimilarity. What we found is that when we selected different plot size from these two forests, the Macajaco and Barrio Colorado Island, when we compared it to our Paleocene sites, and we compare them to our Maastrichtian sites, we saw that consistently the Paleocene rainforests were far more similar to Neotropical forests than those of the Maastrichtian. So this shows us that, even at a low level, there is a lot of overlap. So the same family composition that we see in these tropical rainforests, we already see in the Paleocene, but not in the Maastrichtian. And this shows us that it's not just a difference, or these pre and post-extinction rainforests, they didn't just differ in the amount of, or the proportion of flowering plants. They also differ in the composition of these flowering plants. And lastly, one of the things that we were able to look at was herbivory, paleoherbivory. And that's because insects, as they feed, they induce these scar reactions on leaves that are actually very nicely preserved, and we can identify in the fossil record. And because insects have various ways in which they can eat, we have a very wide spectrum of these functional types that we describe as damage types. So discrete trace fossils that are actually reflecting how insects are functionally feeding on leaves. And we can identify them, we can differentiate them from mechanical damage. So it's actually a very powerful tool that allows us to quantify herbivory and compare across different fossil sites. So what we did is that in all of our leaves, or in most of our leaves, we looked for all these different types of damage. We morphotyped them using presence, absence, using a standard guide. And we compared how similar were the different damages that we found across host plant species. And what we find here, this is again a dissimilarity index. So 0 is very low dissimilarity or high similarity. What we found is that in Bogota and Cerrejón, there's overall a very high similarity of leaf damage across host plants, meaning that basically all insects were eating on all plants, and they were eating the same way. They were leaving the same types of trace damages across all host plants. Whereas in the Guadalajara flora, which is the Maastrichtian one, we find that there is a very low similarity, meaning that insects were being quite selective. Not all plants were showing the same types of damage or the same chewing marks, meaning that there was to some extent some sort of host specificity by the insect communities. And that host specificity changed completely, or this distribution changed completely when we moved into the Paleocene. So when we put all this together, we saw that, pre-extinction, post-extinction, we see that, both types of forests are rainforests, but we go from having a community that is mixed, that is made up by ferns, conifers, flowering plants, to one that is fully dominated by angiosperms. We see that, we go from a forest that had open canopies to one that had closed canopies. We see host specialized herbivory and then widespread herbivory. And we see that legumes and other typically tropical tree families, or families that are currently dominating in the tropics, are also found in after the extinction. So basically, all this is telling us that forests, rainforests, neotropical rainforests, they're ecological services, which are highly associated, or intrinsically associated with the abundance of flowering plants, broadleaf, to leaf area index. So the amount of leaves that we find, per unit area, that is ultimately describing, that transpiration capacity of the rainforest. And it's also ultimately translated into productivity that we see in rainforests today. So what we're seeing is that those ecological services of neotropical rainforests, they actually appeared after the end-Cretaceous extinction. And then the question is why? Why was there such a drastic ecological and taxonomical turnover, especially considering the climate didn't change much? And considering that, flowering plants before the extinction, they were already pretty diverse. They were already pretty, functionally and taxonomically diverse. So why weren't they forming these closed rainforests, flowering plant dominated rainforests before the extinction? So, we came up with three ideas that are more, there are ecological, they're not mutually exclusive, and they're not the only ideas out there. But one reason could be due to, differences in disturbance. And this has to do with large herbivores, large dinosaurs, and how we know from modern ecosystems that these large herbivores are actually able to shape their surroundings. So this we see with elephants in the African savanna. We also see with, in Pleistocene Park, where the reintroduction of large mammals is actually changing boreal forests to boreal savanna-like environments. And even though we don't know for sure what the kind of like the dinosaur community or large vertebrate community looked like in the Maastrichtian, we do know that in the northern part of South America, at least by 100 million years ago, we had pretty large sauropods. We had various types of other dinosaurs, such as belisarids. And these are just like very punctual examples of the dinosaurs we had there. So it could be that, we have these large dinosaurs roaming around, and they could be actually, disturbing the forest to the extent that they were actually keeping the forest open, keeping open canopies. Once they became extinct, this disturbance pressure could have been eliminated, and perhaps that is why rainforests were actually able to kind of just flourish and grow, forming closed canopies. Another idea is due to selective extinction. So, as I mentioned, most of the conifers that we find in the Maastrichtian rainforests are monkey puzzle trees, or relatives to monkey puzzle trees. And overall, this is a little bit of an extrapolation from modern ecology of this group, and they have a pretty narrow ecological range, meaning that they're all large trees. They grow quite slowly, and we know from the fossil record that in these types of clades that have a narrow ecological range, they're more prone to extinction. So it could be that these plants, we know they became regionally extinct. Could be that once they were hit more severely by the extinction. And once they became regionally extinct, this niche, so their position in the upper canopy, just became available for flowering plants to take over. And lastly, one idea could be related to soil fertilization. So both due to a precise effect of tsunami-related events, so debris that was taken in from oceanic deposits that is associated to the tsunami wave caused by the impact, all the ashfall that came from regional fires. And that could have been the kind of like fertilization, immediate fertilization could have given a jump start for flowering plants, which we know they thrive in high nutrient soils. So kind of to start taking over initially. And then once they became established, we see that this abundance and diversity in legumes in Paleocene rainforests could have further contributed to kind of keeping a high nutrient content and high nutrient cycling in tropical rainforests. And that would have further favored angiosperms, flowering plants, to be dominant over ferns and over other conifers. And lastly, just to mention, one of the key things that we found is that we saw that 45% was the regional extinction rate that we saw in these South American rainforests in the end-Cretaceous. And surprisingly, when we look at current rates of deforestation in the Amazon, it's expected that by 2050, about 50% of Amazonian plant species are projected to be endangered. So it's really astonishing to think that something that number, around 50% extinction rate, is what we find during one of the most catastrophic moments in Earth's history. And due to human impact and habitat destruction, that's kind of where we're going. So just something to think about. And with that, I would like to thank you and take any questions if there's any.
Yeah, thank you, Monica, for that talk. That was a lot of interesting work and a lot of effort and hard work just compiling all of that data set. So thank you for sharing that with us. Okay.
Yes, needless to say, a lot of people were involved. A lot of co-authors, a lot of undergrads, a lot of volunteers have been involved in this.
Yeah. Great. So I think we can get started with questions. And as always, feel free to drop in your questions in the chat box. And we can also get started. We'll get started with some questions that have come in. I'll also copy the question and put it in the chat box if my voice breaks or you just want to read the question again yourself. So the first one is, how much of the herbivory signal do you think is taphonomic versus a true signal?
So that's a very interesting question, an interesting point. So with herbivory and leaf damage in general, it's a field that started something about 20 years ago, and as more people are joining in, there's a lot more discussion in terms of what are we actually seeing. So the thing is, because we see that there are patterns, right? Overall, when we look at herbivory, and because there are patterns that are biologically consistent, we see that there is a signal there. To what extent are we what part of that leaf damage are we missing in the fossil record due to taphonomy? Probably a lot. And nonetheless, because we're comparing very similar deposits in this case, we see that very similar deposits, very similar types of fossilization, kind of the same level of detail. That's why we make these comparisons. But it's something that we definitely have to think about, especially when we're comparing different types of fossils, different types of fossilization, different types of environments. different time periods, and especially when we're comparing it to, modern herbivory. So there's a lot of work that still has to be there. Nonetheless, there's a researcher, Sandra Snapchat. Can't remember how to pronounce her last name. She's doing a lot of really, really great work in like putting down, okay, what are all the biases that we have to think about, and to what extent can we really take this quantifiable thing and make inferences about it.
Yeah, cool. So almost like trying to quantify the uncertainty in the signals that you're seeing. That's cool stuff. Cool. Okay, so we have another question that I've also put in the chat. So the question is, those leaf fossils are incredibly well preserved, and it is really awesome that you've been able to get both Maastrichtian and Paleocene samples and compared them to modern ecosystems. What a fantastic data set. I was wondering whether you have any sense of how the community changed between Paleocene to modern. Was it a more gradual shift, or were there similar step changes like the KPJ boundary?
Yeah, that's a good question, and that's something that we have to keep on. We have to fill in the Paleocene to the recent gap in terms of fossils, but we're working on that. What we see from the pollen record is that there are definitely, in terms of diversity, because we can't really get into composition too much with the pollen record. Too many plant families, too many pollen grains, which we have no affinities to. And what we see is that, at the Paleocene-Eocene boundary, there's a spike in diversity in the tropics. But then there's also a decline, and there's another spike associated to the Miocene climatic optimum. So I do think that, to get to today's composition, I think there has been a lot of change. And it's going to be very interesting, as we keep on finding fossils that allow us to identify specific clades of tropical plants, to see how the assembly of what we see today actually came to be. I think we're going to be seeing moments in which there's massive radiation. We don't know why. And then definitely extinction, which is something really cool, because as paleontologists, we're the only ones who can really see that. But you cannot really get that information from, molecular phylogenies.
Yeah, no, definitely. Okay, and there is a question that just came through. Very cool work, thank you. Do you have any ideas for testing the 3 hypotheses you put forth in the last few slides?
Yeah, so one of the things that we that have been kind of working on and will keep on working has to do with that nitrogen idea, so the nitrogen fixation idea. There should be, and our preliminary data shows that there seems to be a difference in like nitrogen isotopic composition in paleosols of before and after the extinction. So that's one way to tackle it, but it's not too straightforward, because nitrogen cycle is as complicated as it is now. Putting it into paleosols is going to be tricky, but that's one way of testing it, because the isotopic signature of nitrogen that gets fixed by bacteria is very, very different and should have a very pronounced effect on the whole nitrogen pool. So that's kind of where we're leaning towards. In terms of disturbance, it would be great to know what megafauna lived in the Maastrichtian, in the tropics, but dinosaurs are quite hard to find, but they're, the fossils are there. We need to find them. And in terms of that selective extinction, that's kind of a tricky one. Because I don't really see too much how we could possibly test that. But if anybody has any ideas, I'd be happy to collaborate.
Yep, there's always more research to be done.
Yeah.
Cool. Okay. So we have another question. What was the procedure for checking for diagenesis while measuring the leaf cuticle delta C13 values?
That's a good question. Overall, what we did, I mean, some of these cuticles we also examined for, like specific compounds. So when we looked at specific compounds and we kind of saw the range of alkanes that we were able to retrieve from fossil leaves, and how different were they from modern leaves. That's one way we checked for diagenesis, and we found that the preservation was very, very good. So some of the longest compounds that we typically, that are also the most abundant, longest alkanes that are typically also the most abundant in cuticles, modern cuticles. We were able to find most of them and in large quantities. So we know that the isotopic signal, the carbon isotopic signal, was mostly from those alkanes.
Okay. So in line with the sort of staying in the realm of isotopes and trace elements, another question was: is it possible to glean more information from other element chemistry in leaves?
Yeah, I think so. Yeah, that's something I'm working on, and it has to do with the hydrogen and oxygen in leaf waxes, and it's also something that the nitty gritty, it can get a lot into modeling. So I don't know to what extent we could actually pull information out of there, but there should be some information on transpiration rates. So enrichment of leaf water just due to transpiration, that should be reflected in leaf waxes. And so right now at the Smithsonian Tropical Research Institute with Klaus Winter and some other plant ecologists and physiologists, we're kind of doing greenhouse experiments, growing plants under high CO2 and high temperature, to kind of induce a very distinct difference in water use, and testing whether we can actually see a signal of those differences in water use in hydrogen in leaf waxes. So we'll see. That was pre-pandemic work, so still working on that.
Yeah, that's pretty interesting stuff. So like parallel to like CO2 increasing in the greenhouse, you're also increasing temperature and transpiration to see the effects. That's cool. Okay, so there's another question that was just put in the chat. Great to see all this research coming together. If you wanted to learn more about neotropical forest evolution, but from a zoological perspective, are there any localities in Colombia that could have higher preservation potential for small to medium-sized animals than El Cerrejón? I'm also thinking about the potential influence of legume diversification on mammal diversification that was hypothesized in another recent paper on faunas and floras from the Paleocene of Colorado, for example.
Yeah, so there's so in the Bogota Formation, so one of these fossil floras, not with the leaves, but in those same sort of deposits, we find a lot of we find some marsupials, so some early mammals. And that is kind of work in progress. I know nothing about, vertebrate paleontology, but I do know that there are mammals there. The problem again is that, the sites where we can look for these fossils are very, very limited. So there are some, very specific spots in Colombia that are on the drier side, either because they're in intermontane valleys, or just because due to regional differences, and there is good rock exposure, but they're very, very, punctual, and not only geographically, but also, in time, in the rocks that we find there. But we have been looking at some other Paleocene formations. So I'm just going to say names that mean nothing to you, but like Socha Formation and Sama Formation that are also, Paleocene, Eocene, and age. And we're constantly looking to see, if we can find some other interesting fossils, not just from plants, but animals as well.
Cool. So there are some, but sort of spread and preservation is always the question. Okay, so we have one last question, and if anyone has any last questions that they'd like to get in, please do so. Otherwise, this is going to be our last question. And the question is, what kinds of data do you think are missing from the paleo leaf assemblages? Are there specific groups that are just not found in the fossil record, even though they should be there? What would your ideal preservation environment be to get the best snapshot of an ancient forest?
Yeah, so one thing that we like in the last 2 years we've been collecting, we've been looking for mesofossils, and we actually found this assemblage of like microseeds. These are like millimeter and a couple hundred micrometer sized seeds. And those are giving us a completely different perspective to what we see in the leaves. So we're seeing more, like those typical tropical families that are not necessarily represented in leaf assemblages, either because they don't produce too many leaves, or because they're not very abundant, but they do produce a lot of seeds. So we're now seeing, we're looking at this other different type of plant organs and different type of preservation in order to get a better idea to what was the understory like. What was the forest understory like? What was, what were these plants that were probably more associated to either lakes or running water? Because a lot of what we found some seeds of Cyclanthaceae, which is, I don't know what the common name of that family is, but it's this sort of, palm-looking family, tropical, and we had never seen fossils of that. It has a very, very sparse and very poorly documented fossil record, because these plants are just, little understory, shrubby-looking things. You just produce a few large leaves and that's it. So I guess the question is, we need different types of assemblages. We need leaf assemblages. We need seed assemblages. We need permineralized things would be awesome as well. But it's always hard, you need a bit of luck to find all these things kind of in the same spot or spanning the same time period.
Yeah. Thank you again. Yeah, that was a great answers to all the questions that people had. And thank you again thank you everybody for joining us. Please fill out the weekly feedback form. The link should again be in the chat so we can learn a bit more about who attended to this seminar. Next week, July 20th, we will have a talk by Catherine Davis, who's at Yale University, and she'll be talking about reconstructing paleo oxygen minimum zones from the shells of low oxygen tolerant planktic foraminifera. And it is time for a break before tea time. Monica will be sticking around and chatting with us more in terms of just if people have questions, want to stay back and say hi to Monica, ask questions that they didn't ask during the chat. During the talk, this is the time to do that. We will have a 2-minute break, so feel free to turn off your video and mute yourself. And then after the 2 minutes, we'll all get back together and have a tea time.